La bienvenida a todos ustedes a esta segunda serie de los seminarios Fronteras en Sistemática, Evolución y Biodiversidad del Instituto de Biología. Al igual que el año pasado, tenemos a un grupo de investigadores de calidad internacional que nos van a hablar de diferentes temas que tienen que ver en la sistemática en su sentido más amplio, en temas que son relevantes para toda nuestra comunidad y que realmente la elección de los ponentes que vamos a tener derivó directamente de las elecciones que ustedes, como comunidad del Instituto de Biología, propusieron. Los invito a que disfruten estas charlas y no solamente la charla, sino que aprovechen la oportunidad del tiempo que generosamente los ponentes nos están brindando posterior a la charla para conversar con ustedes o con sus estudiantes o con diferentes miembros de nuestra comunidad. Sin más, damos paso al inicio de esta primera sesión de la segunda serie de los seminarios en Fronteras en Sistemática, Evolución y Biodiversidad del Instituto de Biología.
Muchas gracias a todos, Ulises, a la doctora Susana y por supuesto a nuestra ponente del día de hoy, la doctora Catherine, que le agradezco mucho haber aceptado esta invitación para abrir los seminarios de fronteras de esta sesión 2022. Yo voy a tener el agrado de dar una pequeña presentación de la doctora Catherine y enseguida la doctora va a empezar con un seminario. Les recuerdo que el seminario va a ser en inglés. Así que a toda la gente que nos está viendo pueden hacer sus comentarios tanto en español como en inglés en el chat y nosotros le vamos a dar lectura al final. Entonces, brevemente les quiero contar que la doctora Catherine es una científica líder que trabaja en las unidades de investigación de biodiversidad y biología de la conservación y ecología evolutiva espacial en el Instituto Federal Suizo para la investigación en bosques, nieve y paisajes. Ella recibió tanto su maestría como su doctorado en la Universidad de Missouri en San Luis y de 2003 a 2017 fue profesora asistente y asociada y titular en el Departamento de Ecología y Evolución en Stony Brook, en Nueva York. Posteriormente, bueno, ella también realizó una estancia postdoctoral, vale mucho la pena decirlo, en la Universidad de Berkeley, en California. En cuanto a sus intereses, es realmente interesante y muy apasionante lo que ella realiza, ya que ella estudia cómo la disposición espaciotemporal de los hábitats influye en la diversidad biológica. Y sus preguntas de investigación son sumamente atractivas, como por ejemplo, ¿por qué las montañas tienen una biodiversidad extraordinaria? O también cuál es la importancia de la partición de los nichos en el mantenimiento de la diversidad biológica. Cabe decir, y lo van a ver en la plática, que también está muy interesada en el ensamble de comunidades, y también cómo influye la historia climática de las regiones en los patrones que observamos en la actualidad. Ahora, para abordar estas preguntas, ella integra una variedad de campos como la ecología, la biología evolutiva, la biogeografía, la climatología, la geología incluso. Y gran parte de su trabajo en la actualidad se centra en el papel de las interacciones planta-colibrí, que es justamente lo que vamos a escuchar el día de hoy, en la generación y el mantenimiento de una alta diversidad en el trópico, en regiones tropicales y muy en particular en el neotrópico, como lo verán. También trabaja en muchos en otros sistemas y preguntas, y muy recientemente se ha involucrado con plantas de montaña en la región de Europa. Alguien, bueno, como un servidor que también está muy interesado en modelos predictivos de la biodiversidad, la doctora Catherine es muy conocida, reconocida por sus análisis de modelos estadísticos para describir la distribución de las especies. De hecho, creo que todos los que estamos de alguna forma involucrados en este campo, sin duda hemos leído muchos de los artículos de la doctora Catherine. Y de hecho, en muchos de sus artículos ha buscado unir la ecología y la biología evolutiva para alcanzar un mejor entendimiento de los procesos que impulsan los patrones de la diversidad de especies. Como ven, es realmente atractivo todo el sistema que está trabajando la doctora Catherine, y estoy seguro que será de amplio interés para nuestra comunidad. Sin más, yo solamente quiero agradecer nuevamente a la doctora Catherine. Catherine, thank you very much for being here with us. And so you have about 45 minutes, and we will see you in the next few minutes. Thank you very much.
Gracias.
Lázaro, can you share the screen, please?
So Lázaro, I will just tell you next each time, right? Well, I'll just start, I guess. It's an absolute
Yeah, I'm sorry. Just
no,
1 minute. I'm just trying to put the presentation on.
Okay, perfect. I was just gonna say thank you. Perfect.
Okay.
Excellent. So I'd like to start by really thanking all of you for your invitation. It's really exciting for me to speak in the UNAM because it's sort of where I started my career. I did my PhD in Mexico. I loved it there. I worked in Las Tuxtlas with toucans and habitat fragmentation, and it was probably the most wonderful place to start one's academic career. So to come back now and speak with you is really, really an honor to me, and I really look forward to meeting you guys individually, and I'm happy to meet with whoever would like to speak with me. So today's talk is called Patterns and Processes Across Scales, a case study with Ecuadorian hummingbirds. But really, it's going to end up being a talk about resources and the role of resources in structuring hummingbird assemblages. Next, please. So I'll have these 4 parts. I'll introduce a little bit about hummingbirds and the system. We'll talk about biogeographic patterns of niche partitioning, and then how we did some experiments to try to understand what those meant at a local scale. And finally, how sort of this resource partitioning influences hummingbird energetics. So all the way from broad biogeographic patterns down to the individual hummingbird using the landscape to better understand the processes and the mechanisms behind the maintenance of diversity. Next. 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 Oops. Okay, so here you go. This is the backbone of the hummingbird clade. I guess many of you are familiar with this. The oldest ancestor of hummingbirds that's sort of living now comes from the lowlands of South America. And the oldest fossil actually comes from Germany. How it got to South America is still a bit of a mystery. But in South America, hummingbirds have a lowland origin from tropical wet forests. Several clades moved into high elevations, including the coquettes and the brilliants in South America, which will be what I talk about today. Next, please. They're widely distributed. There's 360 some species of hummingbirds. You can see they live in deserts and rainforests, with particular hotspots in the mountains, and in particular in the Andes Mountains of South America. Next, please. Hummingbirds have tremendous morphological variation. You can see here the wide variety of colors of bills, anything from Ensiphera, the sword-billed hummingbird, to, say, Phaeothornis, which has a slightly curved bill and tends to use heliconia flowers, which are from the banana family. You can see a big range of morphology, and this diversity has also fascinated scientists for years. Next, please. Hummingbirds rely on nectar. They also do eat insects, but they rely on nectar. And back in the 80s and even earlier, people like Caldwell and Feinsinger were sort of suggesting that specific hummingbirds would use specific flowers. And you can see that depicted on the left there. But there's also just a tremendous variation of flowers that hummingbirds use. And for example, in the Gesneraceae, they're sort of responsible, the idea is, for much of the evolution of the Gesneraceae family. So as they move to be hummingbird pollinated, you have more and more species. So there's also a lot of fascinating stuff with coevolution that I won't talk about today. Next, please. And finally, they certainly eat a lot. So they consume 2 to 5 times their weight in nectar every day. So that's the equivalent of a reasonable sized person eating 300 hamburgers. So you can just imagine the sort of challenge they have to meet these energy requirements. Next, please. So that's my very brief introduction to hummingbirds. And you'll have to excuse me, I'm a bit thirsty and I have an alcohol-free beer, which I might take a sip of periodically because it's evening in Europe. Next, please. Okay, so when we think about a local assemblage and what's sort of the diversity in a given place, we often have this sort of idea of these different filters that act at different spatial and temporal scales. So you have a bunch of species, and there's some sort of abiotic filter. Some species can exist under the physiological sort of constraints imposed by this abiotic filter. And then you might have a biotic filter, say other species that they compete with, or resources that they need in particular to occur in that local assemblage. Next, please. So I started out looking at this a few years ago, and I'll just introduce this as sort of a framing of this talk. This was sort of the concept of community phylogenetics. And the idea is that maybe we could learn something about the relatedness of the co-occurring species in a given place, and maybe that would give us some hints about the mechanisms structuring a community. So if you sort of look on the left side, if you have species, if some sort of trait conservatism is assumed, so that means that related species have similar traits. So if you have trait conservatism and you have phylogenetic evenness, then your local community, which is that circle there, you would have a bunch of species that would have really different traits. And the idea is this would be consistent with biotic interactions and niche partitioning. In contrast, if you have environmental filtering, you would have a bunch of species co-occurring with the same trait. And this would be seen through a pattern of phylogenetic clustering. So the idea is you can look at the phylogeny and make a hypothesis about the mechanism that might be influencing those co-occurring species. Next, please. And so that's what we tried to start with. And our first papers were looking at all species that occur in Ecuador, and then asking what was in the local assemblage. Next, please. And so when no filters were considered at all, we found that in high elevations, there were a bunch of closely related species co-occurring, and that this is consistent with a strong environmental filtering influencing local community composition. And as I hinted at earlier, some clades have moved into the high elevations in the Andes, notably the brilliants and the coquettes, and the Andes tend to host species that are somehow related to each other. Next, please. But this is a species pool of everything in Ecuador. So instead, what if we just ask, of the species that could live in high elevations or could live in, given these environmental conditions, do we still see overdispersion? Do we still see a signature of related species co-occurring? Next, please. Next, please. Oh, time for a sip of beer, non-alcoholic beer. The slide should have moved forward. Sorry, something must be, oh, perfect. Okay, so now we're going to consider the abiotic filter and only those species that can co-occur and ask, who's in the local assemblage? Next. And the answer is, when you consider elevation, which is a very, very strong filter in the Andes, all of a sudden you see distantly related species co-occurring. So this is actually consistent with biotic interactions shaping the local communities through limiting similarity. So we have two filters. One is a biotic filter when we abiotic filter, when we consider that abiotic filter and say who can live here, really, based on their environmental tolerances, then we get a completely different pattern. Next, please. So these results suggest that multiple filters influence local assemblages, which is great. But what's really going on in the local assemblage? So looking at a phylogenetic pattern of who's related in a given assemblage allows you to come up with a hypothesis. It's not an answer. So we wanted to go into the local assemblage and actually test this hypothesis with local data. So we went to the Andes of Ecuador, and this was work done by my then graduate student, Ben Weinstein. And we wanted to sort of ask the question, we know that biotic interactions appear to influence species at broad scale. So what is the local scale mechanism behind these patterns? And you can see we worked in Ecuador, little green map there you should be able to find. And we worked across a gradient from 1,500 meters to 2,700 meters. And common along this gradient were 23 species of hummingbirds. Next, please. And we basically wanted to get at the mechanism from how we go from this abiotic filtered species pool to a local assemblage. And you can imagine different mechanisms and different kinds of things that might go on. You might have exploitative competition, and so species, and these aren't mutually exclusive mechanisms, different species using different resources, which would lead to niche partitioning. But you could also have interference competition. So you could have really strong biotic interactions. So maybe every hummingbird wants to use the same resource, but only some are particularly aggressive and can use that resource. And so these are sort of two types of competition that might be important in influencing the local assemblage. Next, please. So how did we test this? So at every 200 meters across this gradient I just described, we put out two hummingbird feeders, one with high sucrose and one with low sucrose. And these values are based on the values of sucrose in this particular forest. So 30% is a lot of sugar in this forest in Ecuador, and 10% isn't so much. And we wanted to measure competition through sort of a measure of how much resource they actually could take or used in a given amount of time. So we measured something called selectivity. So that's the time the hummingbird spent on the high value resource versus the total time it spent on both values, with the idea that selectivity is a balance between the energy expended to access the resource and the energy gained from obtaining the resource. Next, please. And so we might sort of go back to those ideas of competition and make some hypotheses about what we might expect. So species should be less selective when there's a more closely related or morphological similar species present at the feeder. And species should be less selective when there's a more massive species present at the feeder. I haven't really talked so much about weight yet, but there's a lot of behavioral ecology theory that says big species dominate and are most competitive. And then the second kind of evidence we would have would be associated with resources or the niche partitioning aspect. So species should be more selective, or an individual should be more selective as the abundance of the preferred resources increase surrounding the feeder. So why bother going to the feeder if you've got all kinds of resources instead that are resources that you, Mr. Hummingbird, like in the neighborhood? Next, please. This is a video, so please press the magic button. Oops. Okay. If that magic button can't be pressed, this is the basic sort of thing you might see if you go to feeders. You see all these hummingbirds flying at each other, right? And so this is really sort of to suggest that we can't expect interference competition in hummingbirds. We see it at feeders all the time. Next, please. selectivity can also be influenced by exploitative competition or niche partitioning. And this is just an example of some of the many flowers that we recorded across this gradient. Next, please. And it's not only how many resources, but also maybe resources that the hummingbirds prefer. So if we knew which resources hummingbirds used a lot, then we might expect this would even be a better predictor than all resources. Next, please. So we collected data. We now have about 8 years of data, but not all will be analyzed here, across 6 transects. These transects were 1 kilometer long, basically using existing paths in the forest, and they were every 200 meters across this elevational gradient I described. We counted the numbers of flowers, we recorded their abundance, and then we recorded hummingbird visits, both to the feeders that we set up in experiments, as well as to the flowers. Next, please. Okay, so some of you might have gone out in the field and said, this looks like a good flower for a hummingbird. And you really want to see this hummingbird, so you sit there and you sit there. And sometimes you see the hummingbird and sometimes you don't. And so getting data on interactions is really been helped by our use of time-lapse cameras. So these cameras take a photo every second. And so you can imagine there's many, many flowers and many, many feeders, resulting in millions and millions of images. And so now you've got a ton of video or images to sort through to see where's the hummingbird. And so my very clever grad student at the time, Ben Weinstein, came up with a machine learning method to do this. This is called deep motion meerkat. And basically you can go through video and identify the organism that you want. So in this case, we have it trained on finding the hummingbird, and it can find the hummingbird with a better accuracy than the poor undergraduate can, or even me, as I sit through and try not to fall asleep looking at millions and millions of images. And this is an interesting line of research if people want to discuss it, where we're doing further work now with collaborators at the ETH. Next, please. So what comes out? What's the answer? So remember, we have this model. We have our dependent variable. We want to understand selectivity. And the thing that best explains selectivity are the preferred resources. So that's the model that just came out as the best. And when we ask what explains how much time a hummingbird spends on a feeder. Next, please. Okay, so that sort of suggests a really important role of niche partitioning. Not to say that other aspects aren't important, but basically species are more selective. So they only spend time on that high resource feeder as the abundance of preferred resources increases surrounding this feeder. Next, please. Okay, so that's cool. How does all this relate to broad scale patterns of even morphologic spacing and assemblages? So how can we take this information to understand those broader patterns? Next, please. So basically what you see on the left side is just an example of a network, or a way to put a network, to draw a network. You've got flowers, you've got hummingbirds, and you know which flowers are being fed on by which hummingbirds. You know the interaction. And so we basically asked, do species, hummingbird species with similar morphology, or importantly for our study, similar relatedness, use the same flowers? And it appears that they do. So hummingbirds partition resources based on relatedness and based on morphology. Next, please. So hummingbirds are selective. So as the abundance of flower resources increases, they use those specific flower resources. Hummingbirds partition resources based on morphologically conserved morphology. So basically, their similar species are using similar resources. So this results in limiting similarity in resource niches. So basically, species that are related aren't necessarily completely co-occurring in the same place, right? And so that results in even phylogenetic and trait spacing at the biogeographic scales. So this sort of idea that related species use similar resources, they partition their niche. And so as you go along this gradient, related species are sort of spread out across the gradient using their specific resources. And then this mechanism would ultimately lead to a pattern of even phylogenetic or trait spacing. Next, please. So how do we understand how we get this even spacing at the local assemblage? So we looked at the resources. So it seems like resources and the types of resources in a local assemblage are driving, or influencing at least, this broad biogeographic pattern. So we can better understand why we see this evenness, because what we see is that related species use similar resources, indicating that niche partitioning is important, and niche partitioning is acting through related species. Next, please. Okay, so now I'm going to move on to think a little bit more about the mechanism behind this niche partitioning. So honing in a little bit more on the question. Next, please. So everybody, or many people, have thought about morphological matching, say between a bird or an insect part and a flower. So this is nice work done by Maria Magalensi, and she's looking at the corolla length versus bill length. And here you see a strong relationship. So species with similar hummingbird species prefer to use corollas that have the same corolla length as their bill length. So there's a matching between the length of the hummingbird bill and the length of the corolla, giving this positive relationship. And of course, the mechanism behind this is sort of limiting similarity and co-occurrence, right? So the idea is that different hummingbirds are going to match with specific flowers. And so not every hummingbird's using every flower. They're using those that match their bill length. Next, please. But this study and all of the studies of this nature have been done at a single time slot, or basically they summarize information across some long period of time. But we know that resources vary across a year. And so there's some times when there's lots of flowers in the forest and sometimes when there's not. So when you summarize data at a site, you miss this variation. And so this is just an example of the variation in floral resources at these sites that we talked about, starting from 1,300 meters going up to 2,500 meters. And this is a summary of 4 years of data. And you can see there's quite a bit of variation in the amount of resources in the forest. And to me, what's really remarkable is it's actually similar across these 4 years, that this phenology in the tropical system is so consistent, but that's not really part of the story. What we want to know is, does this sort of trait matching mechanism that Maria discovered, is this sort of occurring at all times of the year? So when there's lots of resources or few resources, is the mechanism of trait matching still structuring hummingbird assemblages? Next, please. And so when we look at the resources and how they vary across the year, we can see some interesting patterns. So what I have on the y-axis is density, or basically amount of resource, and on the x-axis is corolla length. So what you can see is that during some times of the year, especially at the lower of the elevations, you have a lot of resources, but all the resources in the forest have small corollas. And this is basically some big blooming bushes that have a ton of small flowers that sort of flood the forest. And so high resource availability is characterized by flowers with short corollas. Next, please. So what does a hummingbird do? Does it then use these short corollas and stop this trait matching? Or does it continue to trait match? Next, please. So you can come up with a plausible explanation almost for each of these scenarios. So now on the x-axis, just to confuse you, I have bill minus corolla. So a small value on the x-axis means that there's strong trait matching, that the bill and corolla are the same length. And on the y-axis, I have visits per day. And then the black line is a time with high resources, a season when there's high resources. And the gray line is when there's low resources. So you might expect that species are really selective when their resources are high, right? The resources they like are high. So when resource availability is high, there's tons of flowers in the forest. They go pick their favorite flower. So you would expect this pattern. Next. But you could get a different pattern. It could be that resources are high, but there's so many resources, they just use the next resource that's available. They don't bother going after their good resource. And then the final option would be C. Next, please. And in this case, you might expect, if trait matching is sort of governing how hummingbirds behave, and particularly because of their bill length or their tongue length, they can really extract flowers best from the ones that match their bills, that regardless of the resource availability in the forest, for the most part, they would continue to use their preferred resources, or there would be trait matching regardless of resource availability. Okay, so now, next slide. What do you think happens? Okay, if you were in a room, I would now ask you to raise your hand for A, B, and C. So I can't see you, but I'm going to pretend that all of you are sitting in your comfortable living room or wherever you are, and you now have an opinion of A, B, or C, and you've raised your virtual hand all by yourself for one of these options. Next, please. So we looked at this using a kind of a cool modeling approach, I think, because one of the issues when you do observations is you often miss information. You have imperfect detection. And this is something commonly thought about in wildlife studies. Like, I want to know if this endangered species lives here. I go look, it's not there. I look again, it's not there. And the third time I go, it's there. And so we want to know what's this detection? Are we actually observing all the possible visits to this particular flower that occurred? So we can develop a hierarchical Bayesian modeling approach where we have two kinds of models. The first is an observation model, and that's basically the probability that you saw something if it occurred. And then is a process model, and this is our hypothesis. So in our hypothesis, we wanted to know, is this trait matching pattern that's common in sort of combined data, is it consistent throughout a year? So as these resources fluctuate. So our process model includes trait matching, so the difference between the trait of the hummingbird and the plant. And it also includes abundance, because one might think a hummingbird is naturally going to visit the most abundant plant, regardless of whether the traits match or not. And so this is the model that we set out to test and to look at. Next, please. And this is just a very simple graphic saying that detection matters. When we look at the interactions on the y-axis and the difference between bill and corolla, so the left-hand side of that is going to be high trait matching. You see that if we didn't consider detection probability, we would see very, very little pattern in trait matching. But when we do, suddenly this pattern comes out. Next, please. And so the first thing we see is that we do see some trait matching. Interaction rate is greater when the bill and corolla length are similar. So basically you have lines that start high and curve down. And I'm showing you a lot of species here, and just because not everything fits the pattern. So when you look in ecology, it's just often messy. And some things, or many of the species here, match this pattern, but some don't. And so all of you hummingbird biologists are thinking, oh yeah, I know what those hummingbirds are doing. Those hummingbirds are sneaking, piercing the base of the flower instead. And so trait matching certainly isn't ubiquitous across all hummingbirds, but we certainly see these patterns in many of them. And this is just an example of the different kinds of patterns we saw. Next slide, please. And that trait matching doesn't vary as a result of resource availability. And we looked at this statistically, but you can also see this from this graph. And so you've got low, medium, and high resources. Those are the different colors of gray. And basically you see the same pattern. You don't see really different, the different grays are all sitting on top of each other. And so basically, regardless of the resources, it seems that hummingbirds, when they're trait matching, they continue to trait match. They continue to use flowers that have corolla lengths that are similar to their bill length. Next, please. And so everybody who's sitting in their virtual living room who picked, you're sitting in your real living room in the virtual talk, who picked C, you got the right answer. Very good. So niche overlap does not vary across different levels of resource availability. Next, please. So what does that mean for tropical resource, for tropical biodiversity? So niche partitioning seems to be mediated by trait matching, not perfectly, but there definitely is a strong signal for that in our system, and others have also found this, especially in diverse tropical systems. And support for hypotheses about the role of specialization and generalization. This is support for the hypothesis that specialization on particular resources is important for the maintenance of high tropical diversity, because throughout the year, this mechanism is occurring. It's not just sort of something you identify when you summarize data across a particular site. Next, please. Okay, so digging even deeper now to the individual level and trying to think about resource variation. And now I'll talk a little bit about hummingbird energetics. And to do this, we're going to have to go to Arizona because we could not get the permit to bring the camera to Ecuador or to bring all the equipment to Ecuador. Something about, who knows. And we're going to start at the Hershaw Creek site. And this site is, well, okay, compared to what we just saw, it looks dry, but this is actually a wet site in southern Arizona. You can see a lot of topographic complexity, a lot of possible microhabitats. Next, please. Especially in comparison to the next site, on the next slide, the Sonoyta Creek slide, which has a similar elevation, but looks much different. It's drier and doesn't have these microhabitats and is much flatter. Next, please. And just to tell you how these sites are different, it turns out that, as you might predict, that the Hershaw site is cooler both in the day and at night than the Sonoyta site, which is the dry site or the flat site. Next, please. And also the flower resources are different at the two sites, especially post-monsoon. So post-monsoon, which is the dry season in this area, you have very, very few flowers at the Sonoyta site, so at this flat site. Next, please. So what's a hummingbird to do? So this is the broad-billed hummingbird that spends its breeding season in North America. It's a little tiny guy, and I'm just going to go into what this hummingbird does in these two sites in terms of resources and energy. So how do you measure energy? How do you know how much energy a bird uses in a day? Well, a common way physiologists use to measure energy is something called doubly labeled water. And so basically, you talk the hummingbird into coming to your feeder. It's usually not so hard with a hummingbird. Next, please. Next, this, so it uses the water. This water is labeled, so the hydrogen's labeled and the oxygen's labeled. Next, please. And then the hummingbird flies away. Oh no, you have to catch the hummingbird. And you have to measure the H2, or the labeled water in its pee. Then it flies around. It uses carbon dioxide. It pees out pneumonia. And so you see you have both H and O labeled, but H and O disappearing at different rates. So O is disappearing both by being peed out and by the hummingbird activity, whereas H is only being peed out. So based on this ratio, next please, you can measure CO2 production. Next please. And it turns out CO2 production is a really good measure of energy expenditure. Next please. So then we use this doubly labeled water method to ask how much energy are hummingbirds using in these different sites at different periods. Next please. And what you can see, sorry, one more. What you can see is that they use quite a bit of energy, and it's really variable how much hummingbird and energy can use. It can use difference of sort of threefold variation in energy use. Next, please. So how does this happen? So we have a little beaker, not a very active hummingbird, to one that seems to be using tons and tons of energy. And we wanted to know how it uses this energy. How does it fill up this energy beaker? Next, please. Next. And so here you go. What do hummingbirds do? Well, they have daytime activities and nighttime activities. And we wanted to quantify all these, look at how they might vary, and see how resources and temperature and environmental variables might influence this variation in energy use that we observed with the doubly labeled water. So we made a completely independent model. Next, please. And this is a model of daily energy expenditure. And basically, you have to add up the amount of energy used by a hummingbird in its basic activities. So basic things like daily energy expenditure is a function of the basal metabolic rate, its thermoregulatory costs, its activity costs, what it does, and its nighttime energy expenditure. And so some of these were from the literature, and some of these we measured. Next, please. So how do you measure this? Well, you do not get the hummingbird to wear a Fitbit. And maybe there's trackers small enough for a hummingbird to actually say what it does all day, but this is tricky. Next, please. Next. So instead, we relied on our good old equipment to measure oxygen consumption, which is a good proxy for energy expenditure. Next, please. And so this is just an example. Actually, this is an energy expenditure, but we did a lot of work on torpor. And we also looked at the oxygen use and the carbon dioxide production of birds that are torpid versus non-torpid. This is actually the infrared camera that we couldn't bring over the border. And you can see on the left side that this hummingbird is hot. And on the right side, this hummingbird is cold. It's in torpor. And so a hummingbird spends really different amounts of energy if it's hot on the left side or cold in torpor. So we thought that temperature could be an important thing influencing the daily energy expenditure. And you can, if you're interested in this topic, my former graduate student Anusha Shankar just published a paper showing that hummingbirds have a really flexible nighttime energy use, and they go into shallow torpor and deep torpor and no torpor. And it's the use of torpor is much more complex than we previously understood. And it's really an interesting, intriguing question. Next, please. And then there's, hummingbirds are great. They behave. They go in these little tubes, and then you can measure how much energy it takes for them, for instance, to fly, right, with the respirometer from the previous slide. But in the literature, it's hard to figure out what is the actual activity budget of a hummingbird. And different studies give you different values. So we looked at a bunch of different sort of ways to measure activity cost, depending on the time the hummingbird spent hovering, flying, and perching. Next, please. And so we could basically, this is our model of daily energy expenditure, which has these different components: nighttime with torpor, nighttime without torpor, daily activity at either a high level or low level. So the left side is a low level of daily activity. So assuming actually they don't hover or fly much and they sit a lot. And the right is a high level of daily activity. And then the basic thermoregulatory costs. And we thought that the nighttime component would have a bigger impact here, but it doesn't. It turns out it's all down to daily activity. And really, it's all down to hovering. Next, please. And you can just do a couple of slides. Yep. Okay. And so here in the back are those doubly labeled water values that we started with. So this was this 24 hours, who knows what the birds do, this is how much energy they use. And on top are different models based on these different behaviors and all these different components of what they do. And you can see that beautifully these things overlap. And you can also see that only the highest activity scenario can explain the energy expenditure, or this doubly labeled water value, insinuated during the monsoon, during the wet monsoon, the early wet period in Sonoida. And so that's pretty interesting. So that suggests that hummingbirds, to sort of use that much energy, are spending 80% of their time doing something active. Next, please. So what explains energy expenditure then? So we looked at two basic things, well, a few basic things, but we looked at the flowers in the environment. We looked at the temperature, especially because that should influence all of these sort of basic physiological measures, and also it should influence their use of torpor at night. And we looked at body mass because, of course, bigger and smaller birds are going to lose heat and need different amounts of energy. And our single best model was, next, lo and behold, the flowers. So the number of flowers in the landscapes was influencing the energy budget of these hummingbirds. Next, please. And so you can sort of imagine what this might look like. So when you have clumped abundant resources, you have decreased activity, you have a lower energy expenditure. Whereas on the right side, when you have rare or scattered resources, you have very high activity and foraging costs. And this is going to lead to a high daily energy expenditure. Next, please. So how does this all fit together? And so basically, the resources, the availability and the types of resources seem to be influencing the number and the types of hummingbirds that can occur, from their behavioral level to how they use a specific landscape, all the way through to broad biogeographic patterns of which hummingbirds are in which communities. And so we've tried to look at these mechanisms all the way from the biogeographic pattern down to the individual pattern. So resource abundance and identity seems to strongly influence hummingbirds, which I guess is sort of predictable, from biogeographic patterns to community composition, to plant-animal interactions, and to this trait matching mechanism, and finally to a short time scale energy allocation. And so while these pieces don't perfectly fit together, they're all starting to tell us a similar picture at different scales about what influences where a hummingbird is, and ultimately what influences broad scale patterns of biogeography or distributions that we see. And so that's sort of the end of this formal talk. And I'll tell you about just briefly the research we're doing now. So we've extended our transect in Ecuador. So now we're going from 700 meters up to 3,400 meters up in the Paramo, just to the Paramo. And then we also have a similar elevational gradient in Costa Rica and an Atlantic forest in Brazil. And we've sampled each of these sites for 2 years, basically using the same method, going out each month. And we're looking at a bunch of things about species turnover, interaction turnover, rewiring, any number of things. How specialized they are, what influences specialization, etc. Many, many papers in progress at the moment. And our question is, will generalities emerge? And so, well, it's a bit far-fetched, maybe, because we only have 3 elevation transects. Our hope is that we can start to ask questions across elevation transects where the species pool is really different. So the speciation, the extinction, the biogeographic history of these regions is really distinct. And ask, do we get the same mechanisms influencing the biodiversity patterns of alpha and beta diversity and networks that we see in Ecuador, also in these other sites? And I think there's lots of promise as we can't go to the field because of COVID. No, that's why I don't have more results now, because it's been sort of really slowed down because of the global pandemic, this research, but we're back on track. Yeah, so the hope is that we'll start to see really interesting patterns emerge by comparing these different places. Next, please. And so that's just our FE international team. So this is some members of when we could meet before COVID, right at the beginning of our project. And this would be future research. Next, please. The current research was funded by a bunch of different generous institutions and really some great field assistants working with us. And we have to thank very much the field sites, because the people there were unbelievably supportive. Next, please. And with that, I'm happy to answer any questions that you might have.
Hello, Catherine.
Hello, how are you?
Good, thank you. Thank you very much for your very exciting talk. I think it's very inspiring for students and colleagues to see a person who is so integrative, because you go from very local scales all the way across to broad scales, from physiology to organismal biology to community ecology. Thank you very much. This is fascinating.
Thank you.
We have some questions from the public. And
it's okay if they're in Spanish. It's fine.
¿Quieres en español mejor?
Yeah, now I'm learning German, so I'm going to end up with some very strange combination of words. So I will keep speaking English, but I definitely can understand Spanish.
Okay, but some questions are in English, actually. But
Either is fine.
Let me say this in Spanish. Si alguien quiere hacer sus preguntas en español, bueno, la doctora Graham habla perfectamente bien el español, me consta, y baila muy bien salsa también. Eso no tiene que ver con la plática, pero son otras de sus cualidades. Let me ask you the question from Hernan Vasquez, one of our colleagues in the bird collection. He says, you have it in the screen. If you pull hummingbirds by phylogenetic group, are there some that fit the H by Asian model better? I'm thinking if Andean clades are a tighter fit compared to those that cross the Andes into North America.
That's a good question. So unfortunately, we don't have this kind of observational data yet for all of the, we can't fit the Bayesian models unless we actually have a certain kind of data, at least the way we're fitting them. And so we don't know this yet. But one of the things we'd like to do in the future, except that you only ever have some small number of information for a small number of birds, but is to actually see if sort of, because what you get out of the Bayesian model is the strength of trait matching. So you're actually trying to predict the probability that a hummingbird uses a flower. So you're predicting the probability of that link. And then you get the mechanism that allowed you to make that prediction. So is it predicted well, and is it predicted by trait matching, or is it predicted by abundance, which that's what we put into our model, but you could put more things in. And we wanted to sort of ask whether there's a phylogenetic signal in these patterns, and what comes out is important for predicting the links for different hummingbirds. But it's tricky, right? Because then we have like maybe this information for 40 hummingbirds, and then it's really hard to to look at that from a phylogenetic perspective correctly. But yeah, I think we could, as we get the information for the other places, we could start to ask that. Thank you.
Thank you, Catherine. Now from Patti Escalante, our bird curator here at the Instituto de Biología. Are there proven cases of altitudinal paraphyletic speciation in hummingbirds?
I think she knows the answer to this question better than I do. Not that I know of, but sorry. I think there's interesting variation across the range, but no, I don't know. Sorry.
Thank you. Ulises Rosas, well, who you know him. Plant species might have different nectar characteristics, which, besides the corolla length, might be selected by the hummingbirds. Are you considering that into account in your preference models?
We haven't yet, because in our first studies we just measured corolla length. Now we've measured and bill length. You could say the same for hummingbirds, right? Is it just their bill length, or is it some combination of traits that influences their behavior on both sides, right? Because you can always look at the networks from the side of the hummingbird making a choice, or from the side of the plant trying to attract different hummingbirds. And yeah, so far we haven't. I think one of the tricky parts is to know what other metrics to measure. So we've measured a lot of traits, and I'd actually love to talk with you more about which other traits you think would be important. So we've measured curvature. But as you get a little bit higher in the Andes anyway, there's not so many curved billed hummingbirds, but there's some, but not that many. We've measured curvature. We've measured that's from the hummingbird perspective, but we've also measured the curvature of the plants, and we've measured the corolla length, and like the ancho, and I mean all these different, like how thick or wide it is in different places. And yeah, how to all put that all together, we don't know yet. Because actually even figuring out how to take the same measures on all these flowers was incredibly difficult, because there's this amazing amount of variation. Yeah, so it's tricky. And then we don't have good information on the nectar characteristics, because getting the nectar from the flowers and enough flowers to be able to say something is incredibly hard.
Okay.
But I think there's more to be done there. And I would love to have the insight from plant ecologists about what we should do.
Great.
Thanks.
Ulises again. Could you comment on the concepts of niche partitioning and resource competition in the context of sessile organisms? I am thinking of the plants that also have to compete for pollinators.
Yeah, so this has been, I like birds, I study birds, and all of a sudden I've had to learn to appreciate plants much, much more. And I think it's really, it's a really interesting question, because if you think a hummingbird has like, it needs to eat something in an hour, right? Otherwise it dies, kind of. Not quite, but it's sort of its time scale is really fast. Whereas a plant, even in a given season, it just needs to be visited by one good pollinator, and that's a whole different time scale. So and it to sort of survive and reproduce, its fitness is based on how many seeds it produces over its lifetime, right? Whereas a hummingbird's fitness is a little bit, if it doesn't make it to the next hour, it dies. And so I think that they have really different strategies, the plants and the hummingbirds, and how those strategies come together to form a network is interesting, and something that's also interesting just from a theoretical perspective. But we now always we call it both sides of the coin, because we're trying to be more educated about the plants. And so whenever we look at a metric like specialization, or when we try to study different characteristics, now we're always looking from the plant perspective and from the hummingbird perspective. And I think plants, sometimes I think they definitely compete for pollinators, but they also probably, especially at high elevations or when resources are scarce, they might actually, the mechanism of facilitation also seems to be quite important from the plant side. And so the plants, in some ways, just want to get the hummingbirds to that patch or that area. And so I think plants are super tricky. So they definitely compete in some cases. So different plants are sort of attracting different pollinators. But at the same time, when there's few resources out there, or those resources don't vary that much in morphology, then it also seems like plants are facilitating each other.
Thank you very much, Catherine. I don't see any more questions in the chat, but I do have one question for you.
Wonderful.
When you learn about all these assemblages,
Mm-hmm.
and how hummingbirds interact among them, but also between hummingbirds and plants, it gives you the impression that it has to be a very long time for this to occur. However, they live in mountains, and mountain systems are very, very dynamic in terms of, climate changes and things like that.
Mm-hmm.
So I keep thinking how this can happen so fast, and how these species interactions, assemblages respond so fast to climatic changes. Now we are in a time of very, very fast global change, not only climate change, but other changes. So knowing all what you know about the system, what can you expect in terms of the response of the whole system to these events?
So I think, so the hummingbird physiology gives us some clues, because if those hummingbirds really are flying around as much as they maybe are, if our models are right, that suggests that you can upregulate your energy quite a bit, maybe more than we expected. That's really a different amount of energy a bird uses. But at some point, you can't upregulate it more, right? And so at some point, there's going to be a limit to whether a hummingbird can exist in a given place. And we certainly see that limit playing out when we go and sort of correlate resource abundance to hummingbirds, how many hummingbirds are there. So there's definitely some things we can learn there. There's some limits. But then I think there's a lot of rewiring. We don't see the rewiring, so that the rewiring is when a hummingbird, can actually use any number of resources. So there's many anecdotal sort of observations of Encifera going to a tiny little flower. So the really long-billed hummingbird visiting a very short corollid flowers. And so certainly I think hummingbirds are quite flexible, and probably plants are maybe also, or maybe they self, I'm not sure. But I think it's hard to know, because it's hard to do these experiments. And so what people do usually is they remove something, or they remove something with some probabilistic reason to remove it from a network. And then they ask, how robust is the network? Or how many of the links are maintained? How does the pattern of this community change if we remove a species? But they never can really account for if that species then changes its behavior. If you remove this species, then now we use something else. And so we've done a little bit of work with these big horrible experiments where you go and you in simpler systems in the high Andes in Ecuador where we cover up the flowers. And we have a new project where we're going to do these experiments where we cover up specific flowers. And then we ask, how much rewiring do you actually see when they're forced to rewire in nature? How much do they actually use different resources? And I think these kinds of studies are kind of needed to give us some bounds, right? We know they can do a lot, right? Hummingbirds are seen in deforested sites. They're seen in, obviously, cities. So some species are really capable of doing a lot. Others aren't. So why are some flexible? Why aren't others? And how flexible are they really? So we hope to start to get at some of these answers with this sort of big experimental approach.
Thank you.
So,
thank you very much,
stay
Catherine.
tuned.
Yeah, some other questions just arrived. So
Perfect.
I think we have time, right, Ulises?
Yep, for me, it's fine.
Okay, thank you. This is from Omar Lagunas. It's in Spanish. Can do you get it?
Yep.
Dice, en otras aves se ha observado
Yeah.
que su distribución altitudinal puede estar condicionada por parásitos transmitidos por vectores.
Yeah.
¿Puede
So,
sostener, Luis?
yeah, so definitely there's been a lot of work done with hummingbirds and sort of parasites because they also have these little mites that live in the vills. So in our study, we weren't doing a lot of mist netting, so we weren't necessarily seeing this just because mist netting, other than to sort of trap them for the physiological measures, mist netting is a really time-consuming event. And I mean, on the other side, in the Andes, they don't seem to move that much, at least at the mid-elevations, the hummingbirds. So they're not doing these sort of big movements, but the distribution. So wait, yeah, don't know. I guess you're not necessarily talking about movement. You're just talking about their distribution.
Right.
Yep. I don't think I know the answer to that.
Yeah, it's a tricky one.
Yeah.
Okay, there's another one from Jorge Nieto, from the Botanical Garden. He says, do hummingbirds store energy as glycogen, fat, etc., during the high resource season to spend it during the low resource season?
Okay, so when we observe hummingbirds, the only time we see any fat storage is during migration. That's what we see. Mostly hummingbirds don't have any fat at all. So they really are interesting species from a physiological perspective because they don't store fat because that comes at the cost of flight, of the ability to fly. So if they're fat, then they don't fly as well and they don't get resources well. So within a season, even as the variation in flowers changes, these hummingbirds are skinny little guys. They're really sort of living on a tightrope. They're on an energy tightrope. And so they certainly, at night, the territorial hummingbirds will feed on a lot of nectar and they store the nectar somewhere in their mouth cavity. And they use that at night sometimes. So I think they have some short-term storage mechanisms, but not in terms of that I know of as glycogen or fat. Certainly when they migrate, they do, but not when they're on their sort of at the sites that we've observed them at. Yeah, so that's a hard question, and that's also why when we look at this sort of nighttime energy expenditure, which isn't even that much of their energy budget, but somehow hummingbirds we've shown that are really flexible in how they use energy at night, and the trade-off between the different types of sleep or shallow torpor or deep torpor are really fascinating. And it could be that hummingbirds are so flexible in what they do just because, every little ounce of energy counts. So I think they're fascinating from a physiological perspective because you don't have to worry so much about what about the stored energy. Yeah.
Well, the next question is from him too, and it's related to this one. Yeah. How do you explain more hummingbirds activity with less resource availability?
I know, so I think there's at some point where they just can't keep going. There's going to be a point where they either move or die, right? They're just not in that place because of the resources. But I think our idea was that pretty much to get the resources that are available, they have to fly a lot. So they still acquire the resources, they just use much more energy acquiring those same resources. But you're right, maybe why don't they just have a little flower and then sit and hang out and do nothing? Yeah, but that's not what we observe. So we sort of think that they are more active because they're going after all those resources to try to get enough food.
Well, there's another question from Lázaro Guevara.
Okay.
Thinking about the need of learning more on specific interactions from global change research, what do you think is the kind of data we collectors should obtain in the field? I mean, beyond specimens and locality data?
Yeah, so I think we should start to monitor interactions. And I think there's clear ways of doing this, and it's harder in the tropics because of just the data with the amount of species. But I think eDNA offers you a lot in terms of monitoring. It has its pros and cons, and I think that combined with remote monitoring systems like cameras or other things, and then more and more machine learning algorithms. So smaller cameras, better cameras. But I mean, a lot of it's just the technology, especially like in machine learning and sort of eDNA, the technology is all there. It's a matter of sort of harnessing it or getting it to work for your system. And then, of course, in the tropics, there's the problem of the libraries. But I think there's a lot more we can do. And also using some sort of surrogate of interaction. So it's a bit harder with pollination. I guess you can grab the stigmas and see who visited, or grab the hummingbirds, which isn't so easy, and see what they did. But other interactions are also easier. So like herbivory, you can somehow get, you can get leaf damage pretty easily, and then pretty soon you can grind up that leaf and say which insects are responsible for that leaf damage. That's not so far-fetched, at least in some places. And so you can start to get some amount of damage and who it is. And then I think it's interesting to actually start to monitor not only the species, but their interactions. But it's tricky.
Thank you very much. And the very last question. This is from Corina Rodriguez. Glad to hear you. I'm interested in biogeography and diversity. I want to know what you think about how to go from observing patterns to discovering mechanisms when experiments are not possible. Oh, that's a good one.
Yes, I think that. Hello, Karina. I think that there you're just going to have to observe across time or across different places. So you have to sort of do a place for time substance, but different places which have variation in the things you think are important. So if you think resources or temperature is important, right? But then it's obviously then a sample size problem because you don't have such a big sample size. So it's hard to get a completely sort of conclusive result. So that can help you get at mechanisms. But I also think looking at a single site over time, or if you can have, so now in the samples we have 2 years of data across this gradient across, say, 12 sites. And so then we can sort of start to ask how things vary across a season, because even within a season, at least with plant-animal interactions, you have very different amounts of resources, right? Which you can then sort of capitalize on and say, okay, well, this is the same hummingbird, so something's controlled for, it's the same site, but now something about the conditions is changing. So I think there's maybe the answer to that is like more careful observations when you sort of target it at places where you have variation in whatever you think the mechanism is. Not sure that's a satisfactory answer. But I mean, experiments are really hard, but I think a few good experiments really give us more confidence in some of our more correlative work that we're generally kind of stuck with.
Thank you very much. I think there are no more questions from the public. And I think Lázaro is coming in, right?
Hello.
Yeah, I'm here. Thank you, Enrique. Thank you, Catherine. It was a very nice talk. It's an amazing talk to start this new series of seminars in our institute. So just I want to let you know that tomorrow we will spend a couple of hours actually with students and academics. So thank you very much, Catherine. We appreciate it.
And as I said before, if you would like to spend longer time, I have no plans until salsa dancing at 9 or 10 o'clock. So you have me as long as you want me. It'd be a pleasure to talk with really anybody who wants to chat. I'm very open.
Thank you so much, Catherine. And also for our students, if you want to talk in Spanish, you can do that. We will try to speak in English or Spanglish tomorrow. So
I can understand
don't be
Spanish
shy.
perfectly,
Yeah,
but it's just
okay.
now some silly German words come out. It's horrible because I have to learn German for visa reasons.
Okay, Catherine, thank you very much. So, Ulises, are you there?
Aquí estamos. Y pues ya para finalizar, le queremos agradecer mucho al doctor Lázaro Guevara y al doctor Enrique Martínez Meyer por haber hecho la propuesta de la doctora Catherine Graham de dar este seminario, ¿no? Entonces, le agradecemos mucho, Catherine. Fue un seminario excelente y le agradecemos mucho que nos haya compartido sus investigaciones. Y muchas gracias, Enrique. Muchas gracias, Lázaro. Finalmente, queremos dar un breve mensaje que el próximo seminario de la serie de Fronteras en Sistemática, Biodiversidad y Evolución será el próximo 22 de febrero y tendremos la presencia del doctor Iker Irizarry de la Universidad de Göttingen. Nos va a dar una charla que se tratará sobre Phylogenomics and Major Evolutionary Transitions. Así que invitamos a la comunidad del Instituto de Biología número 1 a presenciar la charla, pero también que nos hagan llegar su interés por reunirse con el doctor Iker Irizarry a más tardar el día de mañana. De la misma manera como estamos haciendo con la doctora Catherine Graham, vamos a hacer reuniones post-seminario donde vamos a poder tener la oportunidad, tanto académicos como estudiantes, de interactuar con el conferencista. Con esto los dejamos y nos vemos la próxima vez. Y no olviden que los seminarios institucionales del Instituto de Biología se proyectan a través de este canal todos los martes a las 11 de la tarde y dentro de estos seminarios están intercalados los de Fronteras en Sistemática, Biodiversidad y Evolución. Con esto les dejamos el video promocional del próximo seminario y nos despedimos. Muchas gracias.